A few more points of understanding about the roots of plant, but this time with reference to the monocot root. Now we are going to talk about the root of a grass. Say, in the previous case we had talked about a dicot root. For that we had example of a sunflower or a pea plant. Now we are going to consider a root of a grass, and that would be a monocot root. See, the points would be similar, but a few more points we are going to understand. And do remember, again I'm saying that remember the points of differences, because that is where your syllabus puts impetus on. First one was epiblema, as we had discussed. The epiblema over here would be the outermost epidermis and the root hair. So this is the epidermis, pyliferous layer. This happens to be the epidermal layer, the colorless cells, living cells which have protrusions known as root hair for the absorption of water from the soil. Next is many layers, multiple layers with intercellular spaces. Again, they would be parenchymatous cells. This particular part happens to be cortex till the endodermis. This black layer is endodermis. Again over here we'll have layer of suberin, and that would be known as Casparian strips would be present over here. You remember the purpose of that. So we have Casparian strips. We have cortex, which is having colorless cells which are responsible for storage. There are multiple layers. There are intercellular spaces. So the second one is cortex, and we have intercellular spaces. So this is the second part. The third thing, not the third, in the second thing, we have to remember that apart from epidermis and cortex, we have one term that we have to come across in the case of monocot root, that is exodermis. Please remember this, exodermis. Exodermis is what? That when this layer sheds off after some time, at some stage of growth, the monocot root often sheds its epidermal layer, and what happens is that because there has to be a separation from the outer soil or habitat, these cells, they would become cutinized. Okay, that means they would developed cuticle around them. This layer has sort of shed off. So these outer cortical cells would become cutinized and form exodermis. I am repeating it again, that at times exodermis is formed in the case of monocot root when the outer layer, that is the epiblema, it sheds off. So at that part, the cells of cortex, they get cutinized and they form exodermis. Otherwise, the purpose of cortex, the purpose of epidermis is quite similar to that of dicot root. You would find root hair, you would find the parenchymatous cells with intercellular spaces for conduction of water, and at the end you find the parenchymatous cells which are closely packed with least intercellular spaces in the form of layer known as endodermis, which is having Casparian strips which is tuberized so that no water passes through them. So almost all things are similar. Okay, now moving further, till endodermis we had cortex, and you have to remember one more thing over here, that endodermis is the site, or not endodermis, below endodermis in the stele pericycle lies, which is having the parenchymatous cells again, and pericycle is the site for growth of lateral roots. Now you would say that why I am introducing this thing, or is it exclusive to monocot root? No, it is not exclusive to monocot root. Both dicot root and monocot root have the lateral roots arising from the pericycle. Just for ease, so that we don't have that dicot root lecture a long one, I have put this point over here. Okay, this exodermis could also be seen in dicot roots, but we have divided the points in such a way so that we come across new terms in both the lectures. They are not exclusive to the one we are discussing. Exclusive characters, I would tell you what they are, but in the case of lateral roots, they both, in case of dicot and monocot, arise from the pericycle, which is part of stele. That is the third one, and that stele has pericycle, vascular bundles, and the pith, which is similar to the dicot root again. Now one thing that is the point of difference over here is that we had talked about polyarch condition. The The polyarc condition is seen in the case of monocot root steel. As you can see over here, these are xylems and this is phloem. These xylems are 6 pairs, as you can see. As many xylems are there, so many, those much phloems are present over here. So the xylem phloem arrangement is polyarc. So the condition is polyarc. Apart from that, we got introduced to the term exarch and endarch. Over here you can see that the protoxylem again here is towards the endodermis, that means it is towards periphery, and metaxylem is inside. So the condition happens to be exarch again because the reference is made with respect to the protoxylem. So the vascular bundles are exarch. They are polyarch. And the last point that we discuss is pith. And over here, the pith is highly developed. Unlike in the case of monocot or dicot root, pardon me, the dicot root had a very small pith. Over here, the pith is quite conspicuous. You can see it is large and it is having large intercellular spaces where we find the parenchymatous, living parenchymatous cells which are present over here. There is pericycle, and in between the phloem and xylem, there lies conjunctive tissue. As you can see over here, the part I am labeling, this happens to be the conjunctive tissue. Now, what is conjunctive tissue? We had discussed it is the parenchymatous tissue that is present between xylem and phloem. So, this is all we have to understand about the monocot root. Do remember the points of differences again, and the main point of difference, or rather you can say the most important point of difference, is that unlike the vascular bundles, which were 4 in the case of dicot root, we have a polyarc condition over here. Okay, then we had exarch arrangement, which is similar to the dicot root because the protoxylem lies towards outside while metaxylem lies inside. The pith is not that much developed in the case of dicot. Over here we have a huge pith. The cortex is similar, epiblemma is similar, rest all things are similar. Only point of difference is in the steel and the number of vascular bundles which are present. So this is all you have to understand about monocot root. Now one thing you have to keep in mind is that you keep very much in track to the points of differences that we have studied in the case of monocot root and the dicot root.